this now, this has got to go back 20 years when he was still an endocrine resident. The Prairies lays claim to Brent. He's originally from Saskatchewan. I can't remember exactly where, but well, the Prairies lays claim to you, and you've had a stellar career. To McGill, to Twins UK, where you did your PhD in genetics, and have done some outstanding work in population genetics. Lead first author in a Lancet paper and many other high impact papers, and so you've allowed me to ride along with you on some bone projects. But basically, you are a rock star in the genetics world in Canada and internationally. So kudos to you, and you are mentoring some rising stars as well, I can see. And Tien Nguyen is a PhD student at the McGill Quantitative Life Science program. His PhD is focused on polygenic risk scores, which is an area that I've had some collaboration with Brent on. Brent is currently a professor of medicine at McGill and has an enormous interest and track record in human genetics and osteoporosis. So with that, Brent, I think you're visible now, and if you're able to, one of you to share your screen, we'll get going.
Okay, can you hear and see me?
Both. And
okay,
your slide.
okay. Can you see my slide? Okay, great. Bill, I was able to hear things. I'm really sorry. The hospital has a lot of internet issues. I'm linking through my phone, and I'm hoping that this will work. It seems to be working now. I thank you, Bill, very much for that kind introduction. You would think, you would expect, I guess, that I could organize a PowerPoint presentation, but I'm apparently still struggling with that. What Tien Nguyen and I are going to talk about is polygenic risk scores. This is a machine learning group in Manitoba, and we at least I pretend expertise in machine learning. We are users of machine learning. Tien Nguyen is an expert and is actually doing his PhD in quantitative life sciences here at McGill. But I'm going to try and give you a bit of a setup to the problem. Tien Nguyen will take you through the problem, and then we'll share with you what we've been able to use relatively simple machine learning techniques to generate polygenic risk scores. I'm just going to start off by thanking Bill. It's funny that, as Bill mentioned, we've been working together for about 20 years, and Bill has taught me probably more than anybody in the world about osteoporosis epidemiology. And when we came up with this polygenic risk score, Bill actually, I was trying to use it to predict fracture. He said, "Oh, Brent, that doesn't look like it would work very well. Why don't you try to bring it into a screening program?" And so, as hopefully many of you have done, I just followed Bill's advice, and this is the result. This work that I'm going to present is really the result of Bill's suggestions, and then many phone calls from, if I remember correctly, Bill, train trips in Lyon when I was living there, when Bill would guide me through what it is that we're supposed to do. Okay, so the first point that I wanted to make was this one. This is the tallest man in the world and the shortest man in the world. And the difference in height between these two men is about 7 feet or so. This is how we think about genetics in the clinic. So we think about people at the very extremes of the distribution, and here is the distribution of height. And when we see some people who are so obviously unusual, we think, oh, this must be some strange genetic cause. And indeed, both of these men have had their genome sequenced. And when that was done, they identified single base pair changes in their genome, which has led them to be very tall and very short. But this man, who some of you may know, is Sean Bradley. I don't know much about basketball, but he apparently is a really good basketball player. He's awfully tall, and he also had his genome sequenced, much for the same reason. And when that was done, what was found was nothing, which is kind of shocking. There was no single base pair mutation or known large perturbations that made this man so tall. What is probably guessable to most of you is that his parents were really tall. And in fact, the reason why most of you are the height that you are is not because of any single base pair mutation, but because of what is actually thousands of little nudges along the distribution that in the case of Sean Bradley has pushed him all the way to the end. And that's just because he has accumulated almost all of the known common alleles that cause you to be tall. They're common, they have really small effects, and if you have enough of them, you can get really, really tall. Or if you have the amount that you have, that explains to a reasonably large degree the height that you are. And so I'm going to try to use this as an analogy to help you think about how we could use genetics in the clinic, how we could use machine learning techniques in order to be able to decipher the contribution of those really, really, really small effects, but that exist in abundance. I'm going to think about that not at the ends of the distribution, but in the middle of the distribution, really just talking about how we could start to think about using polygenic risk scores within the entire population, just as these 4 people here that you might recognize, at least I recognize from my childhood. So the talk outline is going to be polygenic risk scores. Tian-Wan will talk about the machine learning aspects of that, and then we'll talk about applications to screening programs, and then we'll end with problems. And there's lots of problems with these sorts of approaches. I'm assuming that you can still hear me. Maybe somebody could chat to me if there's a problem, or just put a thumbs up. Okay, great, thank you. Okay, so these are very similar to my math skills. These are our good friends at the National Trust of Scotland, where if you buy 18 months worth of banking for the price of 9, you get 6 months free. And so that's about the extent of my math skills. And I'm going to try to conceptualize polygenic risk scores without using any math. And then Tian-Wan's going to get into the math. So what are polygenic risk scores? Polygenic risk scores just take single nucleotide polymorphisms. So a single nucleotide polymorphism is just a very cumbersome way to say a variation in your DNA at one base pair. And some people in the population have one flavor of that base pair, and some people in the population have the other flavor. And because they're polymorphic, we call them polymorphisms. And there's only one of 2 flavors, and because you have 2 chromosomes, you can either have 0, 1, or 2 of those risk alleles. And so you can bin everybody in the population into one of these 3 bins, and you can say that some people should be taller than others. And because these have small effects, you wouldn't expect them to be very much taller. But you can add in 2 SNPs, and when you do that, now you pull out your distribution a little bit more. You can find the people who have 4 risk alleles, and the people who have none, and the people in the middle. And then you can do that with tens of SNPs, and you can just keep classifying people into counting the number of risk alleles that they have across all of the SNPs that are known to be associated with the trait of interest, in this case, osteoporosis-related trait called bone density. You can get to hundreds of SNPs. And what you can do is you can actually get yourself to thousands of SNPs now, and you can pull out the distribution just of the population for any trait, whether or not that be predisposition to schizophrenia, lung cancer, height, or bone density. And you can start to ask simple questions like, how much variation does this score explain in the population? And in order to do this, what you need is a good understanding of the genome. And this is the human genome in 2005. And what I've done here is I've placed 2 little pins where there were reproducible associations with common disease. And you can see on chromosome 1, there were 2 of them, which is pretty shocking if you think that the American government spent about $2 billion sequencing the human genome, and which was completed with an address on the presidential lawn in 2000. 5 years later, 2, which is pretty paltry if you ask me. But then with the advent of genome-wide association studies, which enabled us to get sample sizes up high and discern these small effects from noise, we started to see lots of associations which were reproducible. When you get to 2020, I've only been able to show a few of the chromosomes on the slide because you can't really see the chromosomes now for all the pins. And this is because much of the human genome has been mapped to its association with common disease. This has led to tremendous investments from industry now who are looking to use this information as a source of causality in humans, because there are very, very few ways in which one can decipher causality in humans, and human genetics is one of them. But we're not going to talk about causality today. We're going to talk about prediction. And in order to think about prediction on a population-wide scale, you need to really think about costs. So is this a technology that we could afford to apply to the population? Well, people tend to think that genetics is expensive, but it's not. It is cheap. So this is Moore's law. This is the cost per sequencing of the genome. Back in 2001, 100 million bucks. Today, we can get a genome sequence for less than $1,000. And the technology that I'm going to discuss today is called genome-wide genotyping, and we can get that done for approximately $40. So for $40, which is a fairly paltry investment, you can capture an enormous amount of variation in your genome, which would render you with millions of single nucleotide polymorphisms. And then you can map those to those little pins that I showed you and begin to understand where people are on the risk distribution from a genetic perspective for a common disease or trait. So that is an introduction to polygenic risk scores. I'm going to then tease you a little bit and tell you a little bit about how we're starting to think about using this in the clinic. And then we'll quickly switch gears to Tianwan, who will tell you some of the machine learning methods that we use in order to decipher these. So this is a paper that we published recently. You can take a look at it later if you're interested. But the first question that we wanted to ask ourselves is, okay, now we have a tool. It's very strongly associated with risk of or changes in bone mineral density. I'm going to talk about bone mineral density. I'm just going to offer you a few sentences on its relevance. So we measure people's density in the clinics because it's a surrogate for the strength of their bones. So it's a density metric. Bill is an international expert in its measurement, so I'm sure I'll say things that are incorrect, but Bill can correct me. But what this is clinically is it's among the more useful metrics by which we can predict someone's risk of fracture. Fracture is very important. It costs billions of dollars per year in Canada, largely because of the rehab costs associated with major osteoporotic fractures. And so the question that we wanted to ask is, how could this be helpful? Well, the first and simplest mathematical way that we wanted to look at this is, what is the variation that's explained in bone mineral density by this score? And in order to put this into context, first I'm going to show you what happens, how much variation can we explain in bone mineral density using three simple metrics. So you can measure these in your clinic at almost no cost with very good accuracy. And we use them all the time. We have no official population-wide screening programs in Canada for osteoporosis. But if you just applied age, sex, and BMI, which is what we tend to do in our clinic, we tend to look for old women who are thin. And we would explain about 4% of the variation in bone mineral density. This is data from UK Biobank. We can add in FRAX risk factors. So what the FRAX score is just a summary collection of clinical risk factors that predict fracture, which Bill has worked on for, well, a couple of decades now. And this is unfair because it's not actually meant to predict bone mineral density. It's used to predict fracture, but it's some surrogate of the clinical risk factors that we use in osteoporosis screening. That improves the variance explained in bone density by a little bit. And what can genetics provide for us? So if we go back to 2008, it provided almost nothing. In fact, I gave talks telling people, forget prediction from genetics. It's not going to work. There's no variance explained. But as sample size increased, and all that enabled us to do was decrease the standard error of the effect size of SNPs on bone mineral density, and therefore allowed us to identify more signal from noise. We started to get at sample sizes of 30,000, variance explained, which was somewhat similar to clinical risk factors, but still not very exciting to me as a clinician, because this I can measure for almost free very quickly, and this would require taking blood, doing the genotyping, etc. So still not ready for prime time. But then when we got up to 150,000 people, we thought, wow, actually this is starting to double. And then when we got up to 450,000, now we're up to 5 times age, sex, and BMI. And we predict that, and then using the machine learning methods that Tianwan is going to explain, we're able to get up to 25% of variance explained. And we predict that using genetic epidemiology methods, which I won't describe, that with an infinite sample size, we could get up to about 36%. And so this is what Tianwan and I use in our lab in order to predict bone mineral density. And this is what I use in my clinic. And what I'm going to try to spend the rest of my talk thinking about is how we could use this information to inform our clinical risk factors. Okay, so this is the setup for the PRS, and now I'm just going to ask Tianwan to add to this his work on describing different methods that he and other people in our group and in other groups around the world have used to generate polygenic risk scores.
Thanks, Brent. Are you able to see my screen right now? Thanks. So as Brent has introduced, the genetic risk prediction is really providing us with a lot of opportunities to understand the disease and also to stratify the population according to each individual's predisposition, genetic predisposition to different disease. So if you have a complex trait, which has contribution of a lot of SNPs, then essentially you can quantify this risk by such a formula where Y is your risk, and you have the contribution of each SNP or each allele denoted by X times their effect size. And if you sum up over all the available SNPs you have, then you can quantify this risk. But the problem is that, as Brent said, there are a lot of SNPs in our human genome. There are actually 6.5 million SNPs with a minor allele frequency above 0.05. And of course, there are a lot more SNPs that are more rare. But with the largest cohort in hand, we have at most 0.5 million samples. So of course you can see this is a high dimensional problem. So that renders us no method to give a linear unbiased predictor of these betas. But fortunately, what we can do as an alternative is to run what we call a GWAS, which is simply a univariate linear regression. You take your response and your genotype at each SNP, and you run the linear regression one by one. So for each of the SNP, you can estimate its marginal contribution towards the trait or towards the disease risk. But of course, you can see this beta J hat estimated in such a way is biased. Statisticians would have no trouble or feel trouble with using this beta in this formula if your features are completely independent. But this is not the case in our human genome. The SNPs, they are actually correlated, which we call linkage disequilibrium. So basically, you have two SNPs that are in LD, in linkage disequilibrium. That means mathematically they are dependent variables. So in that case, say here, if you have four causal signals, four signals contributing to your disease, if you use all of the SNPs, then these correlated SNPs would have roughly the same marginal estimates for the betas. And then, of course, you create a bias towards your final risk prediction. So how can we resolve this problem? There are two commonly used ways. So we want this PRS to be a summation of per allele contribution of the SNP, but we know that they are biased. So the first way is to tune this beta here such that they are less biased. One way to do so is what we call LD-PRED. This is an linkage disequilibrium informed coefficient adjustment method. So basically, if you have correlated SNP, then this method would tune down the effect size of the correlated SNPs, but only retain those are relatively independent. So in such a way, the beta J hat prime can give a less biased predictor. The other way is to manipulate on the M here. So I know there are LD blocks here. I know there are bunches of SNPs that are correlated with each other. So why don't I just select one from each of the bunches? So that is what we call pruning and thresholding. We first imposing an LD thresholding step where we select representative SNPs from each of the blocks. So from each triangle, we select one SNP, and then we look at their p-values. This is a second step called p-value thresholding. So we select the important representative SNPs to put into our final prediction model. So in such a way, we can tune the M here. We do not include redundant information in the hope that we have a better prediction. So I'm going to illustrate this with an example. So say we have a trait of interest, and we have data from the UK Biobank, which is one of the largest cohorts to date. So we can first split this entire data set to a training, a validation, and test set. We can run GWAS, which is univariate regression, on the training set, so that we have the marginal, we have the estimate of the marginal effect size. And then we can select different parameters in different methods. We can construct a score based on these different parameters, and we can evaluate them on the validation set. On the validation set, we can then select the best performing parameter such that we can then construct our most optimal risk score and evaluate its performance on the test set. For example, we recently constructed a polygenic score for adult height. On the very top, this is the unadjusted version of PRS. We have redundant information. We have dependent variables. So it could explain 22.3% of the total variance in the validation set. But we also have the improved predictions using two of the methods mentioned. So the first is by LD-pred. We had different parameters, and we can see with some parameters, the performance would be substantially improved. We also have the pruning and thresholding strategy. So we can see also compared to directly using the raw marginal effect size, we can improve the overall prediction as well. That being said, so some of us came up with the idea that why don't we tune both of the two parameters? We exclude the redundant information, as well as making the estimates less biased. So the one natural way to do so is to perform a lasso. So I assume some of you have already known how it works. So basically, you put a penalty on the L1 norm of the coefficient, such that you have a sparse predictor. Some of the SNPs with less predicted power would have their coefficient set to 0. In such a way, we have less SNPs included, and we have a beta that is re-estimated jointly with other predictors. So they can hopefully become less biased compared to using the marginal statistics. So of course, we can repeat this model selection procedure in the validation set using different lambdas. Lambda is the penalty we put on the L1 norm. So we can select the best performing score corresponding to the best performing lambda. If we add these results to our comparison, we can see that fortunately, the lasso made one large step more on top of the best performing LD score or the pruning and thresholding score. So our best performing score eventually explained 41.8% of the total variance in adult height. So this is almost doubled the performance of the raw score constructed by all the SNPs with all the marginal effect size. So following the same strategy by performing lasso, we also constructed our recent score for predicting the risk of osteoporotic fractures. So we basically followed the same framework. We split the UK album into 3 sets. We performed GWAS here, and then we selected the best performing PRS with the optimal parameters, and then we assessed its performance on the test set. This is what we call a GSOS, or genomic ultrasound speed of sound, which is predictive of the fracture risk. So you can see that compared to individuals with the highest genetic risk, those with the lowest genetic risk has almost 5 to 6-fold decreased risk of having osteoporotic fractures. And this risk stratification is also consistent if you take into account the effect of age. Compared to our common risk factors at the same prevalence level. For example, if we binarize the population into 2 groups, one group has the 10% highest genetic risk, and 90% have the lower genetic risk, then the population at the higher genetic risk would have almost 2-fold increased risk of having the fracture. But with some common risk factors like smoking, 8% of the population would smoke, but this would only confer 1.5-fold increased risk of having fractures. So I won't go into too much detail about the evaluation, but if we include such a genetic risk score into FRAX, a common used tool to screen the population for fracture risk, then we can happily find that this genetic score assisted risk prediction has prominent improvement over just using the FRAX score consisting of only risk factors, clinical risk factors. So a simple summary, how do we build a polygenic risk score? We first split the data into 3 sets. We perform GWAS on the training set, and then we build initial PRS for selection on the training set with different parameters. We can optimize the choice of parameter on the validation set, and eventually we can test it on the test set, and we can do more evaluation and explore ways to incorporate them into clinical usage. So that being said, people may wonder either lasso or other methods, they are really basic machine learning methods. How can people use more advanced machine learning methods? The answer is we can, but in many cases where this has been tested, we found the polygenic risk score, which is a linear combination of the genetic risks, is actually the most, is one of the outperforming scores. If you include other methods that include either nonlinearity or include interaction, that may largely reduce the generalizability. You may, your methods are likely overfitting. So on an independent test, they may have very limited utility. But we hopefully we still see the some promising work recently. So there's a lot of room for improvement and space to explore. So finally, I want to thank my supervisors and all the colleagues in the lab. So thanks very much.
Thank you, Tianwen. I think that that was a great presentation, and hopefully was pitched at a level that for those of you who are interested in these methods may have some questions. So I just wanted to pause here really to get feedback and also pause for questions. As a machine learning group, you know, this hopefully will have been the most exciting part of the talk. And if you have any questions for Tianwen, just go ahead. I will also present, you know, how we have used this polygenic risk score in what may be a more clinically relevant way. But just wanted to pause here. So go ahead if you have any questions.
Yeah, it's Lisa here. I've got a question. Thanks, Tuan. Can you go back to your slide 16 that compares the different methods?
Yes.
That one. Yeah, I was interested in what happens with the regularized regression. So you've got much more variation there, right, in the AUC. Do you have an explanation for that?
So this one is not from our group. This is a recent publication in genetic epidemiology. So I'm not entirely sure how exactly they implemented the regularized regression, but it doesn't seem to work well. That's all what I can see.
Yeah, compared to the other methods, I mean, the mean value is similar, but the variance is quite substantially different. Okay, so no, I was just curious if you had an explanation for that. Thank you.
Thanks.
Other questions from Tianwen? Thank you, Lisa.
Yeah, Rob here. I'll just comment following on Lisa's question. I had the same sort of, hey, that's a surprising figure, this slide 16. I've read some things where, and maybe it's the size of the training set that's critical, but places where logistic regression was performing at least as well and sometimes better than the more intensive machine learning methods. So I'm going to follow up on that one and figure out a bit more about it. But that's a really interesting set of AUCs.
Yeah, I think that's a great point. So I remember their training cohort includes only 7,000 cases plus 7,000 controls. So this is a really small cohort compared to UK Biobank or other large cohorts. So yeah, that may explain the loss of power. Yeah.
Incidentally, great presentation. Thank you. That was a lovely overview.
Thank you.
Hi, this is Razvan Romanescu. That's a very interesting talk. My question is, are you going to come up with a piece of software that does this lasso method applied to just generic polygenic risk scores? Because I think that would be very useful to many people.
Yeah, so one of the hurdles we had is the dimensionality. You can run lasso, but it takes weeks or even months. But there's a package called Big Lasso, which we use in our work. We didn't develop that, but the Big Lasso is really facilitating inclusion of thousands of SNPs at a time with a large number of samples. So that's a good package, Big Lasso.
Just one comment on that, Razvan, is that there's still lots of optimization that needs to happen here. So we can only feed into Big Lasso thousands of SNPs. I'm certain that there's signal amongst millions of SNPs, and we just don't have the computational capacity to be able to deal with that.
Right, that's understood, yeah.
Yeah. Okay, so what I'm going to do now for approximately the next 10 minutes or so is go over how we have used these scores. And these are, what Tianwan showed you is that this is a predictor of a predictor. So clinically, we're trying to predict fracture, but what we used to do in our clinic to predict that fracture is we measure bone mineral density. And therefore, this polygenic risk score predicts a predictor of fracture. And so Bill, when I get stuck in osteoporosis epidemiology, I often phone Manitoba and say, what do you think, Bill? And Bill said, well, Brent, why don't you use this as a predictor, not of fracture, but as a predictor of bone mineral density, and you could incorporate it into the osteoporosis case finding programs, in fact, maybe even screening programs in the future. So I'm going to try to show you what we've done with that. This was a large scale effort with groups around the world where we used 5 different cohorts to explore these ideas, which we think may be relevant to healthcare systems. So all screening programs rely upon a hospital or some healthcare administrative authority screening a population to identify people at risk. They're all sort of structured in the same way. You take a population and you divide people into one of 2 groups. You either reassure them or you recommend some sort of treatment. When I say treatment, this may be a pharmacologic treatment, it may be a lifestyle change, it could be education, it can be anything. And I'm just going to point out that this work here was done by a very talented research associate in our group, Vince Forgetta. And when you push people through a population-based screening program, there's several different steps that generally tend to happen. A large proportion of the population you don't even want to screen, and that's because their baseline risk is not high enough. And often a very good metric for that is age. We don't screen the young girls for breast cancer, obviously. Once you screen them, though, you then divide the population into different risk groups. You could divide them into 5 different risk groups. You could divide them into 2 different risk groups. Here I've just shown 3. And the dirty secret, and the reason why we have very few true population-wide screening programs in Canada is just this. And that is that almost a large proportion of the population that you actually screen, you just tell them that they're okay. And that's because, of course, the population prevalence of any disease is not high, of any single disease. And so unless you have a really helpful thing that you can do, there's no point in screening 100% of your population if you're just going to tell 80% of the people that they're fine. Unless it's very cheap, and unless the consequences are very high, in this case, very expensive osteoporotic fractures. But these screening steps are based upon heritable risk factors. And so Bill's insight was to say, okay, why don't you use your polygenic risk factor here to identify people whom you actually don't have to screen. So what conceptually, I live in a universe which doesn't exist, admittedly, but that universe is a universe where everybody has been genome-wide genotyped. And I very strongly believe that this will happen. It's already happening in multiple healthcare jurisdictions around the world. It's very cheap to do. Everybody gets genome-wide genotyped at least once in their lifetime. This data can then be used for multiple different things. It can be used to predict outcomes. It can be used to identify people who are at the very, very ends of the distribution. But here we're trying to think about, okay, if I could genome-wide genotype everybody in Manitoba, what would I do with that information in this screening program? Well, what you could do is if you're actually measuring bone density and that costs a reasonable amount of money, then you could screen out the people who you genetically predict would have such a good bone mineral density that it's not worthwhile actually measuring the bone density. And you could do this for breast cancer, which has been shown. You can do this for coronary heart disease. You can do this for measurement of lipids, which our lab has several different papers under review right now. But you can think about using this genetic pre-screening step in order to try to decrease the burden of the population screening step by moving a lot of your population into this box. Okay, so that's really what we're trying to do here. The risk is that if you put somebody into this box and you actually measure the risk factor, then they could have ended up in the high risk box. And so you lose sensitivity to identify people who should have had an intervention. And if you do that, then that can be very dangerous for your population. And it really depends upon, you know, how many false negatives you're willing to accept. And so what we did is we took, I'm just going to go over this really briefly. We took 5 different osteoporosis cohorts around the world. These had various different characteristics. Some of them were just older women, some of them were just older men, some of them were middle-aged people, some of them were quite relatively young people in their middle age. And what we did was we applied what has been proposed to be a population-based screening strategy within the UK, and that's because the Brits are probably the closest to implementing population-wide screening for osteoporosis of any major country. And these are called the NOG guidelines. And we just asked, if you introduce this genetic pre-screening step prior to moving people through the NOG guidance, which will cause most people to have a bone mineral density measured, what happens to your sensitivity? So you don't want to have a large loss of sensitivity, but can you remove people from the population who don't need to have their bone mineral density measured? And if you do, how much of a savings could you have? So these are the results. The first and most important thing is, does sensitivity decrease? So standard of care to identify people who should have an osteoporosis screen based upon their absolute risk is very high. And GSOS-enabled screening decreases this marginally. It does importantly decrease it, but it's 94%. And so you're still picking up the vast majority of people in whom you would recommend an intervention. Specificity remains the same if you enable this GSOS screening step. The number of subjects that require the clinical risk factor visit drops quite importantly by 37%. So that's the relative decrease in the number of people who actually need to go and see their doctor to be screened. And then the number of people who actually need to have a bone mineral density test done decreases by 41%. So I've glossed over many details. I'd be happy to go into this as much as you would like. But what this suggests is that you can start in the context of osteoporosis screening to think about how you can use genetic prediction to decrease the number of people that you need to screen. And if you're a healthcare administrator, this then makes an osteoporosis screening program much more attractive if the costs of genotyping are offset by the cost savings of not doing the osteoporosis screening. The concept here is that you wouldn't use these genotypes just for osteoporosis screening. You would use them for breast cancer screening. You would use them for coronary heart disease screening. You could use them for lipid screening. And so that one single investment of $40 in a research context could be applied to many different areas to enable screening programs to decrease the burden of the number of people who actually need to be screened. So we're hoping to get a lot of questions and criticism, which we're always open to. But that's really a summary of our talk. And just prior to going through the summary, I'll outline some problems. But I just want to pause here and point out that genetics explains an important proportion of variance in bone density. I think that's fairly indisputable now. This is going to be captured to some degree through GSOS, and really it's the machine learning folks who will increase that variance explained. We have a long way to go. We believe we can get up to at least 35%, and that at least we can start to think about how we can use such polygenic risk scores to improve screening programs. But let's talk about the problems for a minute, and I just have 3 slides on this. The first problem is, will patients actually do this? So if you offer this to patients, will they do anything about it? And I see patients every Tuesday and Wednesday afternoon, and I can see Bill's laughing because I know he sees a lot of patients. And it's always surprised me that you spend so much time thinking about risk modeling and thinking about communicating risk, and then you impart your lovely information, which you've spent years thinking about, to a patient, and they sort of shrug it off and say, "Bah, I don't care." So that's a big hurdle. The other is a really big hurdle that is really taken up a lot of Tianwan's time, is that the dirty secret of these polygenic risk scores is that they've been developed on European data. By that I mean European ancestry data. So most of the very large genome-wide genotype cohorts that we have around the world, like the Canadian Longitudinal Study of Aging, these are like very predominantly European ancestry. And when we translate these risk scores to non-European ancestries, their performance attenuates, sometimes dramatically. And so Tianwan is leading a worldwide initiative to be able to translate these to Chinese ancestry, Japanese ancestry, South Indian ancestry, and African American ancestry. And he's assembled the largest collection of cohorts around the world of non-European ancestry to be able to do that. And he could talk for really hours about some of the struggles that he's had using different machine learning techniques to try to translate these to these other populations. But I'm just going to say that that's a problem. The other problem is these guys. So the men and women who are physicians are a very recalcitrant group of people. They generally don't like change. This is a nice quote from Eric Topol, which really, I think, demonstrates that I still use a pen and paper when I go to my clinic to write my notes. And presenting polygenic risk scores to physicians is going to be a big challenge. Not something that's probably going to be solved by machine learning, but something that is a really real world barrier in trying to implement such technologies in our clinic. So to wrap up, hopefully I've convinced you, and Tianwan more convincingly has shown that this is the future. These are very few people within the distribution. The polygenic contribution to disease explains tremendously more variants than rare genetic variants of large effect, and that's only because they're rare. Genetics moving forward in the clinic in terms of prediction will really hinge upon polygenic risk scores, in my opinion. That's not to say that they will definitely make it to the clinic, but they will definitely make it in some form. And for most people, they'll be much more hopeful than rare genetic variants of large effect. Polygenic risk scores can explain a large proportion of the variants. The cost to generate these scores is low, and it's dramatically low. And that's really, I want this to be a take-home message. We need to think creatively about how to incorporate such scores into clinical practice, particularly in screening programs. Cost-effectiveness studies are desperately needed in this field. And I'll just close by thanking some people. And the UK Biobank, is an amazing resource, and I would encourage you to get access to that data. It's really in a huge data set which can provide a lot of information. We work very closely with the FRAX team, particularly Nick Harvey, John Canis, and Eugene McCloskey, who taught me a lot about FRAX. We also worked a lot with our statistical genetics group, which is sort of our sister lab here at McGill, and that is Celia Greenwood's group. Audrey Duran has made a very large contribution and is working very actively on non-European ancestry polygenic risk scores. Obviously Bill has made a very large contribution to this work, and I really appreciate him inviting us to give you this talk, and hopefully we'll have enough time now to get your feedback and thoughts upon this program. So I'll stop there. So thank you very much for taking up some of your Thursday morning.
Well, thanks to both of you. And first, I'm going to correct some statements. I think Brent is projecting a lot of his good ideas onto me. So thank you for that, Brent. But no, I don't deserve near the credit that you give me. I'm really glad that you touched on the ancestry bias issue, because that has been very much a theme that's part of visits with Howard Bachner and the JAMA series. I'm not sure if people are listening to his editorials through JAMA on machine learning, but he just had one recently on curating machine learning algorithms that I would encourage you all to look at and listen to. And they were again talking about the ancestry bias that can easily get baked into some of these things. So Tianwan, good luck on that. I think that will be very, very interesting. I'm going to let other people ask questions in our remaining time, and if there's a pause, then I'll pipe up. But please, others, take advantage of this, as this is a rare opportunity. Brent and Tianwan are really leaders in this field.
Maxime, nice to see you.
Nice to see you, Brent. Thanks for your talk, and Tianwan as well. It was very interesting. I have a question. So you mentioned how polygenic risk scores explain a good proportion of variants. And I'm wondering if you see epigenetics sort of helping explain some of the other variants and maybe hopefully reduce the drop in sensitivity.
Okay, so we have spent a fair bit of time on epigenetics, but of course, one of the main problems with epigenetics is tissue type. So we've only used whole blood. We have used this. These findings are a bit old, but what we found using Illumina arrays for ascertainment of epigenetic modifications of DNA methylation sites across a consortium that we published several years ago. It was a reasonable sample size. It was about 12,000 people. We found nothing.
Okay.
With a capital N, and worked for years to try to find something, but we never found anything. We published a negative results study. And that's not to say that epigenetic modifications don't influence things. It's just that moving forward, if you think that we're interested in bone, that would require probably biopsying people's bone. And that, or at least trying to find some way to use sort of blood-borne cells where the methylation modification is similar to what we see in bone. So you could think maybe osteoclasts would be similar to some of the white blood cell lines. So some work to be done there, but it's interesting. We find nothing. Most of our colleagues have found nothing. So I don't have a lot of hope there, but that doesn't mean that it won't move. The needle won't move in the future, as you've seen, just by increasing sample size and improving technologies. With genetic base pair variants, we've gone from like trivial amounts of variance explained to like more than 5 times what we can measure in the clinic.
Quick follow-up to that, Brent. The cost on the genotyping has dramatically gone down. It was extremely expensive to do the epigenetic methylation analyses. What's happened to the cost on that?
So the technologies have improved a lot. So now you can do whole genome bisulfite sequencing. So we actually just paid for just one set of osteoblasts to undergo whole genome bisulfite sequencing. And that's sort of the gold standard now. It actually renders a lot of data. It was really expensive though. It was about $800 for one experiment. So that doesn't mean that it won't decrease in the future. It just means that it's, you know, at least it's a cost prohibitive technology now to get up to tractable sample sizes.
Right.
Yeah.
Thanks.
I just one more comment before I'm going to have to run, but thank you again, both of you. Really fun to see these methods being developed and deployed. Important to note those changes you saw in the performance of the sort of clinical usefulness of these of the extra information looks similar to the kind of thing that you get when you simply adjust the threshold for sensitivity and specificity. It was, you know, that was really what was driving this, and it's interesting. It might be interesting to for a skeptic, like someone who's sort of professionally skeptic about this stuff like me, to show that you can't achieve similar changes in the clinical performance, the clinical usefulness sort of measures, by simply adjusting the thresholds of the original information. So just one thought there, but also I applaud that it feels like you're able to do this work, find many hundreds, thousands even of SNPs and individual pieces of information that are useful, and you're not getting too hung up about the interpretation of those individual SNPs, because I think that's been one of the things that has sort of held back when we went from 5 to 10 to 20 to 200 to 2,000 features in our models. We were, I mean, interpretation of those patterns is very important and probably speaks to mechanism and basic science and all those sorts of things, but letting go of that a little bit to allow yourself to have those much richer collections of feature sets, and just saying, well, look guys, we're not exactly sure why, we're not exactly sure how, but it is working. I think that's a really important thing to move the needle upwards on those performance markers.
Yeah, you know, I find it jaw-dropping when when the machine learning community comes to me and says, yeah, but you don't understand how that works. And I thought that was really important to you guys. As a clinician, I mean, maybe Bill feels differently, but most of the tests that I order, I don't really understand. I really don't. Like, I understand their sensitivity, I understand their specificity, I understand their clinical performance characteristics. But to pretend that most clinicians understand the mechanisms of all of the tests that they order is a fallacy. I don't know why the machine learning community hangs themselves up on this, because clinicians don't. And there are many clinicians who do, and there's many academic clinicians who do, but your average, when you go and see your GP, ask them to explain to you how PSA works. You know, it's a, so I think that we need to let go of that. These are just predictors. That's all they are. And we have an enormous investment in function. We spend a lot of time working on function arising from genetics, but that's not the purpose here. The purpose here is just simply prediction.
I found that on Monday I'm a predictor, and on Tuesdays I'm an explainer.
It just depends on your question, though. You know, in really, you, as you said, you have to let go of this. Yeah. Go ahead, Tian-Wan.
No.
No. Okay. Yeah. So so your other question, Robert, was about sensitivity and specificity, and the threshold which you select. So as I tried to demonstrate, this is a normally distributed score, and so you can set that sensitivity and specificity wherever you want. So if you're a healthcare system that really wants to make that $40 one-time genetic investment, and then you would like to prevent most of your tests, you can decrease the number of tests that you do dramatically, and you can drop your sensitivity to identify people who should have an intervention dramatically. So that's a values question in my mind, where you set the threshold for sensitivity and specificity, which is a trade-off in the number of tests that you receive. We actually have a graph in the supplement of that paper, which shows you know, how that scales across different levels of the threshold. And I didn't show that here. It's actually a bit complicated to explain, but hopefully it would address your question.
Yeah, it was really just a feeling that someone who was thinking, well, couldn't you achieve similar improvements in clinical performance simply by changing the threshold? You know, it'd be nice to show that the best you can, the best utility can be extracted at a certain threshold, and then the extra information you've got through your methods is above and beyond that even.
Yeah.
Okay, we're at the top of the hour, and I think I'll once again thank Brent and Tian-Wan for this wonderful presentation, kicking us off to a great new academic year for the Machine Learning Interest Group. And once again, plug for speakers, contact Lisa, myself, or Allison. We're looking forward to hearing from you. Thanks all.
Thank you, everybody.
Thank you, Brent. Thank you, Tian-Wan.